Welcome everybody to our ISSS and ISTA webinar series today. I have two guests here for our topic, the seed journey from laboratory insights through production to agricultural impact. The two people who are here are Lucas Aru and Ganesh Kumar, and I will introduce them a little bit later. First of all, let us have some more information. This webinar, like always, is recorded, and we will make it available on the ISTA YouTube channel. And there will be links, I think, from both the websites from ISSS and ISTA. So you can easily find it and have a look at it again. And if you want to ask questions for the question and answer session later, there is a question box where you could put your questions, and we will read these questions a little later. Thank you very much for attending, and on behalf of ISSS and ISTA, I welcome you. Our first speaker is Lucas Aru. He's a PhD candidate from La Trobe University in Melbourne, Australia. He's specializing in single-cell genomics of seed dormancy and germination. His research uses cutting-edge technologies to explore how plant genomes wake up, a nice word, during germination, focusing on understanding how individual cells transition from dormant to germinating states in Arabidopsis seeds using advanced single-cell transcriptomics and epigenomics. Now, I hope everybody understood what we just said, but I think Lucas will have a few words for that. He's working under Professor Matthew G. Lucy at the La Trobe Institute of Sustainable Agriculture. His work aims to optimize germination processes in crops for improved agricultural yields in the context of climate change and food security. His research has contributed to significant publications, including a Nature Plants paper last year on single-cell transcriptional states during seed germination, and a book chapter on plant single-cell technology, which was published this year, and recent advances in seed omics experts' view in the Journal of Experimental Botany also this year. He currently serves as a co-editor of Phytogen, the Australian Society for Plant Scientists newsletter. I welcome you, Lucas, and the floor is yours, and we are looking forward to a nice presentation. Thank you.
Hello, everybody. Thank you, Andreas, for the nice introduction. So I hope you can hear me properly and you can see my screen properly as well. So my talk today will be about what we do in our lab, decoding seed biology through single-cell omic analysis. So it might be quite heavy as a title, but the goal of my talk is really to show you some key points and what we are currently working on to give answers to breeders and for industry. Because the main question of our research, the fundamental research in the lab, is really to understand how this developmental process, this key developmental process happens thanks to which genes and which transcription factor, how this transition happened between the seed development towards germination, towards the vegetative growth and the crop growth. So our goal is to identify the key genes, to give tools for the breeder to fine-tune this gene and the developmental process to optimize the yield at the end in the field. So of course, this is a seed webinar, but I think I like to mention that every time, that seeds is really the world's main food reserve. So that's why I think seed research is very important, because seeds account for 70% of food production through the productions of various resources, rice, cotton, wheat, all the grapes for all the wine, as well all the food for the livestock, and so on and so on. And seed dormancy really helps to maximize the yield of these resources by helping the plant to survive unfavorable environmental conditions until it has sufficient water to germinate evenly. And once the seed dormancy has been released progressively, the genome must start the expression of its genes one by one, and then progressively all together during early germination, and then allow this transition from seed development to vegetative growth. So I could really summarize my research with these four keywords: seed germination, um, really the genome regulation of seed germination. And I am working on this using Arabidopsis thaliana, which is the lab rat of plant biology, if I can say. And I'm doing this not on the bulk view, but really in each individual cell, hence the single cell resolution here. So here I'm talking about genome regulation. So we are really at the genomic level. But I think it's important to remember that in one cell, there is a lot of omics layer. And a lot of people now in plant biology are trying to investigate each omic layers. So we have the organization of the genome with epigenomics. We have genomics with the genes and the expression of the genes, which leads us to transcriptomic and so on and so on. So I won't go into much detail here, but if you're interested to know about all the omics technology and all these different layers that have been used in seed research, I will let you go read this article here. And in our lab, we are really focusing on these three layers, I would say, from epigenomic to transcriptomic, using different technique I will mention today in this presentation. So far I really used the word genomic, but I didn't really use the word single cell. So what is single cell genomics? Well, genomics is the study of genes and their expression, and single cell because it is at the single cell resolution, one cell by one cell. So if you imagine a song, the genome with several notes, that would be the genes. If an entire orchestra play this song, we will hear it as a whole in the same way as with bulk sequencing. However, single-cell sequencing will make it possible to identify which individual musician is playing which part of the music, that is, which cell expressed which genes. And I think plant biologists really um, um, really jumped into single-cell omics in the last 10 years, since 2007 in the first bulk sequencing. So that's a lot of commercial technology that have been progressively applied in the last 10 years. So it's really technical. So today I'll just focus on the main principle and what we're using in the lab. But once again, if you like to have a look of what is done in the global plant world and have an overview, I can let you go read this. So the general experimental procedure we use for single cell technology in the lab is quite straightforward, as would say, as when everything is optimized. So at first, we really have our tissue. So here our seeds, and then we need to separate each cells. So there is two ways of doing it. Either you can work on protoplast or you can work on nuclei. So the idea is to extract this protoplast of this nuclei, and for the sake of the presentation, I will just use the word cell here. Then you need to purify this extraction to keep pure nuclei without cellular debris. You can do this by doing washing or using cell sorting. And then you need to really separate the nuclei one by one by doing what we call compartmentalization. And then once you take back each individual nuclei, in this nuclei, if you do single cell transcriptomic, you do the reverse transcription to go back to cDNA, you amplify all this, then you prepare everything for sequencing. But then when you reach the sequencing, you have all the reads all together. But let's say the advantage of single cell technologies here in transcriptomic is that for each individual DNA molecule at the end, you can trace it back thanks to these three barcodes. So you have the sample barcodes because we have different samples. Then you have the cells barcode, which will tell you that these molecules of DNA is coming from this cell in particular. And then you have the unique molecular identifier in green here, which is unique to each individual cDNA coming from each individual transcript mRNA. As a matter of example, I mentioned nuclei extraction and or protoplast generation. So right now we're mainly working on nuclei after working on protoplast. And I think this part is really what takes us the longest time in the lab, because this nuclei extraction purification is really stage dependent, tissue dependent, developmental stage dependent, and so on and so on. So we have to optimize it every time we work on something new. So it really has to be pretty nuclei with no deterioration in the membrane, because if not, all the component within the nuclei, including the transcript, will just flow away and you will not get anything in the sequencing. So then once you have all your nuclei, as I mentioned, you need to do a compartmentalization, which means separating each nucleus using what we call droplet-based microfluidic. So we use the technology by 10x Genomics, if some of you know this company. So what we have is that we have our purified nuclei here all together, and then we have gel beads. And in the chamber of this machine, this microfluidic chamber, the bead will come on one side, and on the other side you can see maybe the nuclei here, up one nuclei and another one. And then like this, by what we call a complete gel bead at the end, you'll have the nucleus, each individual nucleus that is barcoded. And I mentioned barcoded here, it's because on the beads, there is plenty of barcode that will be able to attach all the transcript, all the mRNA. So once we have all these individual nucleus on the bead, then we will, and after all the mRNA bind to the beads, to the barcodes, we will break the bead, we'll do library prep, prepare for sequencing, and then this is the fun part of analyzing the data. So this is really the workflow we're using in the lab once we optimize the extraction of the nuclei. And why really are we using this technology on seeds? Well, we already knew that there is significant chromatin reorganization during seed development and germination. As you can see here from the seed maturation, the end of the seed development, when the seeds are soaking in water towards germination, we knew that at the end of the seed development, we had progressively the compaction of the chromatin, so reorganization and recompaction of the organization of the genome, which then started to go down and go down, which led to an increase in the size of the nuclei. So this really showed us from this publication that we had something happening here at the epigenomic level. And one previous study from our lab confirmed that at the transcriptomic level. So this study from the lab showed by doing bulk sequencing at that time that from the dry seed towards the end of the germination, here you can see all the genes, they're really separated in three groups, some that were downregulated progressively towards germination, some that were transient upregulated, and some that were upregulated only during germination, but not before. So this view is really a bulk view, a global view, it's the global orchestra view. But we really wanted to know in each cell which genes is expressed, because here it's all the cells. And we wanted to know in which tissue precisely these sets of genes is expressed, or these sets of genes. So this is when we use single cells technology for the first time, which led to this publication last year. And I think today I'll just present you some key items and key findings from this publication. So the goal was really to measure the gene expression in each individual cell during the germination. So we used 3 time points, 12, 24, and 48. We released the embryo out of the seed coats and then performed protoplast, and then we launched the nuclei onto the beads up to sequencing. And what we find is that between the beginning of the germination and at the end, we find that there is, at the end, the germinations or it gives rise to many cell states with distant function. By cell states, I mean all these groups of cells here with different color. the cell state would be that one color represents a group of cells with the same transcriptomes, with the same set of transcript. For example, in this representation at 48 hours, you can see that this group of cells here in green really represent the mesophile and has a distinct transcriptome from the epidermis in brown, for example. And actually, we represented the data like this, but when we work on single cells, we like to use this kind of representation, which is a UMAP. So here, each dot will represent one individual cell, and a group of color will represent cells that share a common transcriptome, a similar set of transcript. So, and in this group of transcript, then we manually went through the literature to identify genes that were known to be expressed. For example, in blue, we find genes that were expressed in the cortex. This is how we can then match this group of cells represent the cortex, this one represents the columella, the root cap, and so on, which allow us to identify all the genes that are specifically expressed in the columella or in the columella and in the radicle, etc., etc. And in this whole data set, then we focused on transcript that gives rise to transcription factor, because transcription factor are the proteins who will bind on the DNA to regulate the gene expression. So we selected a few one and did a germination assay. We worked on mutant, and we saw that the mutation of key transcription factor that we identified in our single cell data set delay germination. So here you can see in black the wild type ecotype of our plant, and here all the mutant, and you can see that actually germination is faster here. And then we also looked at what's going on once the gene is not in the genotype, and we saw that a lot of genes were upregulated, downregulated. So we really saw that completely removing one gene could completely change the transcriptome. So this is one key finding, but I think the main finding of this paper is that cells go through a common transcriptional cell state at 12 hours during germination. So even though at the end we have different cell states, a lot of different group of cells with different transcriptome, all those cells that have different transcripts at the end, they share all the same transcript at 12 hours during germination, knowing that there is no cell division during the germination process. So that's really, I think that's mind-blowing, because it means we have really a transcriptional remodeling here. We have really evolution of the transcriptome without cell division. So this gave us a lot of information from 12 hours to 48 hours, but now we really want to know what's going on before, at early germination, and even before that, after the seed development. So now we are really focusing on what's going on during the release of the seed dormancy, because that's a really key process to have a homogeneous harvest at the end of the season. So in this project, we're also using a single cells technology, but on two batch of seeds that are dormant and seeds that are releasing dormancy. And the goal at the end is to do the same thing as the previous project, to identify the groups of cells that share a common transcriptome, and maybe identify cells that are specific to the release of the dormancy. Well, spoiler alert, it looks like we're going in a good direction, because seeds releasing dormancy exhibit distinct cell states. So I could also say that as that the release of dormancy display a specific group of transcript. So in these two representation, on the left is all the nuclei coming from the dormancy sample, and on the right all the nuclei coming from the sample that are releasing the dormancy. And you can clearly see that we have, in this different group here, we have more nuclei. And you can also see that here, you can see that some of our group, if we mixed, no matter the sample is dormant or release of dormancy, we can see that some group are really in the majority made of nuclei that come from the release of dormancy. So So we really saw that there is an impact and a change in the transcriptome when the seeds decide to release the dormancy. So now that we have some ideas, we are still working on this project. We really want to link what is going on after at the release of the dormancy and before the germination. So we are trying to focus at the early germination stages, around very, very early time point between 0 hours and 12 hours. That's our second current project. We want to know how does the genome wake up, and basically we want to know which genes are expressed the first, which one decides it's time to germinate and go through this amazing process. And to do so, we want to add another omic layer. So we're still working on transcriptomic to identify the gene expression, but we also want to identify the chromatin organization. And both of these data sets are actually coming from one nuclei, which means that by using one nuclei in the same process as before with the beads, we can have two sets of data sets with the same barcode as before. We'll have the transcript, and with the second set of barcode here, we'll be able to get only the open regions of the chromatin, which are the ones that are accessible to transcription factor to bind and then regulate the gene expression. So one nuclei, two data sets. And here I just want to show you maybe some preliminary data we have on the epigenomic side. So for example, what this kind of data can show us is that we can identify specific regions that are open and that are open to accessible to transcription factor. So for example, we confirm that the NPL8 region becomes accessible at the beginning of the germination, and we know that this NPL8 region and this gene is a master regulator involved in the dormancy. So as you can see here, and still in this kind of representation, all the blue cells is the latest time point, and in the pink one, it's the earliest time point. And when we look at the region, we want to know if this region is present in our data set. We can see that it's mainly present in the latest time points. So this region become progressively accessible for then the genes to be regulated and expressed. So I think that's really interesting. This gave us idea, but so far we don't really have visual validation of where it is located precisely in the seed. And this is where comes spatial transcriptomic. So spatial transcriptomic visualize where are expressed the key genes you want to locate and key transcription factor, key transcript, that gives rise to key transcription factor. So to do this with spatial transcriptomic, we really map the gene activity in tissue sample by identifying the spatial localization within the tissue of the transcript of where the gene is expressed. So basically, if I take again my orchestra example, I could be able to say, yep, these cells here is expressing these green genes, this one here, the yellow genes, and so on and so on. And there is different kind of spatial transcriptomic technology that we're using in the lab. There's two main category of this technique on the market as of today. There is the sequencing based. So same principle with the sequencing, but you will put your seeds here in our case, and we do tissue section. We'll very thinly do a section of the seeds, put it on the slide. We'll have barcodes in the same way as before. The size of the barcode will be the size of one cells approximately. Then we will do an image to have a record. We'll permeabilize the tissue. The transcript, the mRNA will go on the barcode, bind the transcript, and then we go towards sequencing. And at the end, you can say, yes, indeed, this purple genes here was located here in this part of the seed, and the green one here. And we are currently working on developing that on seeds in plants. And I do think the most important here is the tissue section, because you have to do one single layer of cells to achieve the single cell resolution. So here, a bit of example of photo we took recently when we tried to optimize this. So this is some ideas of what it could get to do a nice tissue section, and then at the end, to allocate the precise location of this gene was expressed in these cells, we need to do some data programming and try to import some machine learning to match our tissue specificity. And at the end, we are really able to identify almost all the cells in our tissue section, which is really optimistic at the end to say, oh, yeah, this gene was really expressed in these cells here or in these cells. Then we also apply another kind of technique in the lab, which is imaging based, so without sequencing, but in that case, they use confocal microscopy. So same principle with the slides, but here this is with barcodes. So you will design a barcode that is specific to some transcript, because the previous method, you can target all the genome and all the genes, but here it's to validate, you want to see the location of this specific transcript of this specific one. So then you add the probe, this will bind to the transcript you want to locate, you look at it, you take an image, you wash all this, and you add new probes, and at the end you can look at now up to like a few hundred different genes. So it's two sets of technology, but they all required sections of seeds, and if we really wanted to have a global view in a native spatial context, so we optimize a technique that allows us to do this, but in whole man tissue in 3D, and of course in seeds. So a few examples of some results we have of transcripts that we know are located either everywhere or specifically in some regions of the seeds. As you can see here, the green dots represent one gene, and the red dots, sorry, another gene, and then you can even see some colocalization with yellowish orangish dots, and that's really, really powerful technique because you achieve the cellular resolution, but we also achieve the subcellular resolution. So here, for example, it's a gene that we know is involved in the cotyledon. So you can see the upper layer of the cell at the cotyledon here. And if I stop the video, I assume we can see there's a bit of background, of course, not the best image, but we can see some purple dot that represent the genes here at different location within the cell. So I think that's really the power of this method. So I do hope that wasn't too heavy. I really wanted to show you all the different stuff we are applying in seeds for seeds biology, and in the hope of giving tools to breeders in the future. But I do think the main question we're trying to answer in the lab is, how does the seed genomes wake up from the release of the dormancy to start the germination, which allow the transition towards the vegetative states, to give answer to breeders, to then fine-tune all this with key genes we will identify, and then optimize the yield and have a better agriculture. So that was a quick overview. We are also working, and we have optimized the protocol and different analysis pipeline on seeds on other projects. I won't talk about that today, but just to show you the extent of the work we do in our lab in Australia. And on that note, I would like to thank you for your attention, and also all the collaborators in Australia, in the UK, and in Asia, and also the commercial partner, 10x Genomics and Millennium Science. And if you need access to any publication I used as support for my presentation, just shoot me an email. My email address is here. You can also find me on Twitter. I'll be very happy to give you access to any publication you need. Thank you.
Thank you very much. You get a lot of applause and hands up, thumbs up. So, and we have in the moment 2 questions, Lucas, but we're going to ask these questions at the end when Ganesh completed his talk. And now I would like to introduce Ganesh. Ganesh Kumar is an accomplished agricultural scientist with a PhD in crop physiology and over 25 years of seed industry experience at Monsanto and Bayer, plus 7 years in academia. Raised in an agricultural family in India, he specializes in plant and seed physiology, abiotic stress, plant hormones, trait characterization, and seed quality testing. Ganesh's notable achievement including discovering the drought-tolerant gene BS-CSPB, leading to drought guard, which is probably a trademark, I think, the world's first biotech drought product, and co-inventing 5 tissue-specific promoters. He has pioneered seed quality testing technologies and fostered industry academic collaborations Currently, Ganesh serves as Global Seed Physiology Innovation Lead at Bayer, is a Bayer Science Fellow, and Vice Chair of ISTA's Advanced Technology Committee. He is recognized for his collaboration and leadership, strategic vision, commitment to sustainable agriculture innovation, and has received multiple awards, including the Society of Asian Scientists and Engineers Career Achievement Award. Ganesh, I'm looking forward what you are bringing us closer because I like the orchestra, and when I'm next time sitting in an opera house and see an orchestra, I would think about genomes and not the music. Now, up to you to give us other ideas and new ideas. Ganesh, your floor is yours.
Thank you, Andreas. Hope you can hear me okay.
Yes, we can.
Okay, great. Can you see my slide?
You get another yes, we can.
Okay, I can go ahead then. Okay. Thank you very much, Andreas. If there is any technical glitches, let me know. So this is um, wonderful beginning for the today's webinar. Congratulations, Lucas, for the phenomenal work you and your team are doing. Single cell omics technology has such a power to unravel some of these cellular level, the functions that is occurring during the seed germination. So what I'm going to do, as Andreas mentioned, so how we can take this fundamental learning into the the next level of uh, germination and leading to the stand establishment in the field. And more so, I would like to connect the dots on some of the challenges along the way, the journey of seed from, fundamentally right from the lab to the grower's field. And in between the organization involved in delivering this seed, what are some of the aspects they're kind of currently observing, and some of those information I'll be sharing. So again, thank you all for coming today, and let me take in the next 25 to 30 minutes to go through the my part of the presentation. So it will be essentially I'll provide you an outline on introduction about the topic, and then we'll look into some fundamental differences or definitions, and I'll provide you some evidences on some of the observation we are seeing from especially from the dormancy point of view. I will not go into my background. Thanks, Andreas, for covering in detail. This will be in the deck if anyone wants to go through it. So let's come to the topic as such. What is the relevance of understanding the dormancy, seed dormancy, in the broader concept? All of us, like working in plant science, be it an academic institute or or an industry or a seed testing organization, all are working towards how to maximize growers or a farmer's profitability. That is the ultimate, probably the goal for all the effort we are putting in, and by ensuring that we are also helping the global food production, right? So just let's take an example here. When the seed is delivered to the grower or a farmer, the seed carries a lot of information along with it. It's not just a seed. The seed is actually now integrated with a native trait. The native trait could be any disease tolerance or its agronomic superiority. So these are the native traits which are incorporated into the seed, would have really given additional benefit for the seed. And similarly, some of the technology that can come through a transgenic approach, like it could be insect tolerance, it could be herbicide tolerance. These traits are engineered into the seed, and again that comes with a lot of resource and time that is spent on that. And finally, these days we may also see the treatment, that is small molecules, some growth promoters, these chemicals that come on the seed that also help protect the seed when it is in the in a growing condition. So when we take all this, the amount of time, energy, and resource spent on a seed that will now deliver to the growers, the success of that seed depends on how well it is establishes in the field, and germination is the first step towards establishing into a nice stand in the field. So that once we hit the 95% and above germination in the field, that makes it a good crop stand, and that lead to the subsequent development, and then finally the yield, which is most important for the grower, and also the sustainability of agriculture and food production. So with that understanding why it is important, now let's talk about how dormancy can play in this journey of the seed. Fundamentally, I would like to make a distinction here. When we talk about dormancy, it is depending upon depending upon the crop. There are different definitions or different conditions, we may say. For today's presentation, I would like to broadly divide into dormancy as it is understood, right, in the normal existence. It is a seeds that are viable but do not germinate even under favorable condition. This is where we have some of the crops, especially in vegetable crops, we have these conditions called temporarily the seed is unable to germinate, and these are under optimum condition. And there are many reasons for this. This could be physiological dormancy like hormones, or a physical dormancy, sometimes seed coat imposed dormancy. There are several approaches to break those dormancy and then make the seed available for growth and germination. However, when today's topic, I would like to focus on secondary dormancy, where the seeds are able to germinate under favorable condition, right? Say, for example, in warm germ, but becomes dormant under unfavorable condition. It's like sometime we do a vigor test or a stress test. We do a very low vigor in some of the genetics, and this is under stress condition only. So if you put them under normal condition, they germinate fine, no problem. So the main contributor for this appears to be genetics plays a major role, and also it is newly harvested crop. That means you harvest and it comes to the lab just in time. We do see this response. And harvest moisture plays a role. We know that when you harvest at the highest moisture, or in corn, normally some of the crops are raised and produced, harvested at say 35 to 40% moisture, that will have an impact on the secondary dormancy. And also sometimes external stress condition. If they impose a very high strength seed treatment, we do see seeds going into a dormant mode under those conditions. And in some cases, it could be other environmental cues that can also impact it. So what is the impact of this? So when you receive a batch in the lab, and if you test for vigor, and if it doesn't meet the spec, and you know that this is a new crop, then there is always a problem that the release of the product is delayed, because we know the potential vigor of that particular product is more than what actually it is depicted at that point of time. So this is where the business challenge is delays the release decision. We may have to wait few more days to release that batch. That will affect the business. This is exactly the evidence or example I would like to show here. This is one of the batches we tested in April 15th of May 2015. So when you look at this, where at the April time point when we tested, is just harvested. This is in Latin America, and the vigor was 70. And if you allow a month, 30 days, the vigor is raised to 88. That means there's 18% gain in vigor within a matter of 30 days. And if you allow for 2 more weeks, it is attained 92%. So if you use a spec of say 80 or 90 as your that is a good vigor to release the product. When you test on a day one when it's newly harvested, you will be underestimating the potential vigor. That is exactly I meant by the secondary dormancy that is causing because of some intrinsic factors in that particular germplasm make it challenging for the business release decisions. Okay, now we also looked into the what are some of the moisture reason for that. Some of the examples here. Seed moisture has a strong association with ABA. ABA is a dormancy hormone, which is level of ABA regulate the germination in the seed. When we harvested at 40% moisture, we looked at the ABA content is nearly 90 picomole of ABA per gram tissue weight. If anything more than 50 picomole, we have seen there could be some dormancy that can cause delay in germination. So as the seed moisture reduces to 20%, you can see the ABA level naturally reduced in the field growing conditions. So this is exactly what happens when you have high moisture content. The seed comes with high residual ABA, right? So and connecting that to the mechanism, how exactly it operates is ABA content and sensitivity of the tissue to ABA play a crucial role in regulating secondary dormancy. This is a hypothesis built based on some literature knowledge that is already present. And we all know that when the seed imbibes, exactly what Lucas was explaining, the cell state changes happens. So when the seed imbibes, there is also along with that there is a production of oxygen radicals and like hydrogen peroxide, nitrous oxide, and these are the compounds necessary to trigger the downstream activity which is leading to ABA catabolism. And once the ABA is catabolized, that dormancy is releases and ABA biosynthesis is triggered. So then GA to AB, GA biosynthesis triggered ratio, that is what determines the germination process. This is actually occurring in the normal condition. So when we have high level of ABA in the seed, what is some of the treatment that is to break the dormancy, like pre-chill, for example. I'm introducing a term pre-chill. I'll explain to you in a minute what exactly it means. Might trigger during the pre-chill treatment the production of some of these molecules, which will ultimately lead to the catabolism of ABA. I will provide some evidence on the ABA catabolism upon pre-chill in the subsequent slides. And GA3 application, we also demonstrate that if you apply GA externally, we can also overcome the dormancy or a secondary dormancy that is imposed because of the residual ABA. So both these factors, you will see some evidence in going forward. So in order to demonstrate that, we developed this experimental design by comparing various methods for dormancy breaking in corn. And I'm sure these terminologies are familiar, and this approach is familiar to most of the seed testing labs, which are in part of the ISTA and also our other organizations. So now we use a treatment one that is control, that's a vigor test, and then we had a pre-chill where the seeds were imbibed, that means put on a moisture germination media, moist germination media, and subjected to 10 degrees centigrade for 3 days. And then the vigor test is performed. Likewise, we also use GA3 application, 300 and 450 ppm, while the seed is in the germination medium under vigor test, that means under cold conditions. And we also use a dry heat treatment, which is 40 degrees centigrade for 3 days, and allowed for germination through a vigor subjected to vigor test. This is an experimental design, and let's see some results associated with this. So here in this graph, the box plot, I'm showing you the treatment effect. Both pre-chill and GA3 application significantly alleviated the secondary dormancy in this experiment. So the gray box, what you see, that is control. That means it is directly, fresh crop was put onto the vigor test. And you can see around 75-ish, the mean germination, mean vigor. With pre-chill for 3 days, you can see the same set of seed showing 95% vigor. So there is almost 30% jump in vigor. That means that is coming from the pre-chill treatment. And with ABA as well, we can see the improvement in vigor. So the with 450 ppm, we could see some up to 10 to 12% jump in the vigor. However, the heat treatment, which caused negative impact in this case, we did see some reduction, or to marginal reduction in the vigor, or there is no impact on vigor because of the dry heat. So there is a contrasting difference between heat and the pre-chill treatment in overcoming the secondary dormancy in corn. So, and then we looked at, let's break those pre-chill treatment into different duration, like 1 day, 2 day, and 3 day. We wanted to see what is the optimum time frame for the pre-chill treatment in some of the genetics what we're looking at. So what it suggested here is, in this study, we have taken 4 hybrids. 2 are dormant hybrid, that is new crop, freshly harvested, and 2 are non-dormant. These are old seed. These are actually 3 years old seeds. They are absolutely, no reason to believe there is the dormancy in this seed. So in the new crop, what you can see, the blue bar is no pre-chill, the control, and then the green bars, like light green to dark green, you can see there is a systematic increase, a stepwise increase in the vigor as the duration of the pre-chill increases. So from day 1 to day 3 of pre-chill, you can see every day of 10% increase in vigor. This is like a dose response curve. Very, very interesting response, and we see identical response in the 2 hybrids we tested, which are newly harvested. On the contrary, if you look at the non-dormant old seed, there is some very interesting observation we made. One of the hybrids, which is known low vigor type hybrid, we had this is used as a low check, and a high vigor type hybrid, the hybrid 4, we call it high check. So in the low vigor hybrid, we could see there is a marginal increase in vigor upon a pre-chill of 2 day pre-chill, but there is no kind of a dose response what we saw in the new crop, but it's very marginal, which is kind of a perplexing at that point. We were wondering why we are seeing that increase, because it is not, were not expecting the residual ABA in the old seed would have catabolized by the time it was there for the last 3 years. Whereas in the high vigor hybrid, there was no difference, because that is as expected, because it has start with high vigor there. So, and then at the corresponding time point, like 1 day, 2 day, 3 day pre-chill, we also sampled subset of the seeds from the the germinating media for hormonal analysis. And here is the hormonal data along with the vigor. So the blue line here, the vigor data, and you can see the red line, what you see in this graph, that is ABA content, and the dot, the green dot, that is GA3 concentration. So same set what you saw previously, hybrid 1, hybrid 2, and hybrid 3, and hybrid 4. As the pre-chill duration increased, we saw there was increase in the vigor, and you can see the ABA concomitantly decreases as the pre-chill duration increases. So the residual ABA from somewhere near 40 picomole to come down to around 10 picomole. So this is a very nice way to demonstrate when we do the pre-chill treatment biochemically, what happens when the cell is actually, when, as Lucas mentioned, when cell fate is changing. These are some of the changes that happened because the pre-chill kind of triggered the catabolism of ABA in this case. and you can see the pattern in hybrid 2 as well. But the one which is told you like hybrid 3, which I mentioned to you earlier, that had shown some response to preacher, appears to have high residual ABA, which was again surprising because in an older seed, seeing this level of ABA is something unusual. Whether it is de novo synthesized or it is lingering in the seed at the time of harvested wasn't not very clear. But upon pre-chill, we could see there is a significant reduction in that. And if you look at the GA3, on the day 3, there is barely started to accumulate. There is not a whole lot of GA3, but if we had continued like day 4 or 5, we would we would hypothesize that the GA3 would have started accumulating, because still at 10C you don't expect it to germinate on day 3. If you continue to keep it at 10C for about say 10 days, then we can see the radical emerging from that. So this is where the ABA going down and GA3 beginning to synthesize happens at the 3rd day of the pre-chill. And then in addition to ABA, we also looked into few other hormones just to understand, what is their role. Like example here, in the same set of seed sample, we looked into jasmonic acid and also salicylic acid in addition to ABA. For instance, here you can see, the orange line salicylic acid and also the purple line jasmonic acid. Very interestingly, you can see the jasmonic acid, the salicylic acid level was start with very high in all the hybrids we tested. So except for hybrid 3, which is little lower than the other hybrids. But if you look at the the jasmonic acid, the purple line, which is significantly high in the hybrid 3. And if you look at other hybrids, the jasmonic acid is not to that level. They are either less than 100 picomole, where it is almost twofold more in this hybrid. So in addition, what it's indicating is there is an evidence for interplay of specific hormonal profile upon pre-chill treatment in these hybrids. That means a specific genetics has a or a growing condition must have played a role in accumulation of these different hormones, which has influenced the germination pattern of that particular hybrid or a seed batch. So all in all, what is telling us is that genetics have a significant role to play. And here I've just pulled some different hybrids, how they responded. We took about 75 hybrids, and then these are all the older seeds, not the new seed. Just want to see elaborate on the pre-chill response in older seed. So this is not in all genetics. In some genetics, we see a good response. In some genetics, there is no response. For example, for pre-chill, we see low response where less than 10% alteration in the vigor because of the pre-chill treatment. And we see some of them are around 10%. And there are some genetics that give more than 10%, like 20, 30% difference upon pre-chill treatment, even in older seed. So this is to further to understand what must be the underlying reason for this. And if you can pinpoint if there is either jasmonic acid, salicylic acid, or ABA which playing a role, then we can come up with a specific tailor-made solution for those hybrids to manage their the quality when you release it to the growers. So in summary, what we have learned today is by looking into all the evidences I demonstrated, that knowledge may help us to come up with a tailored approaches. Like recognizing that secondary dormancy can impact seed release. That highlights the need for some tailored approach based on where the particular product or a hybrid is grown, what are some of the seed characteristics. This could involve adjusting, the planting strategies or and maybe some treatment to minimize the dormancy issues. And also the genetic component, which I explained, and also the interaction with environmental that needs to be considered. Since the findings indicate that genetics and growing environment play a role and seed dormancy, now the breeders can select germplasm that are more resilient to dormancy issues. So then now they can consider this as one of the metrics needs to be included in their advancement plan or a model, or at least understand what is the risk of certain lines which are having this kind of tendency. Then hormonal management, we know that end of the day we can manage it effectively by altering the ratios of GA to ABA. This interplay knowledge, what we saw, and pre-chill that triggers perturb the system so that we can understand those, the profiles of these hormones, and then probably come up with a hormonal treatment or a management plan. And then I would also think that optimized seed treatment, if you know that some of these molecules can positively, help break the dormancy, that can go as a dormancy breaking method, and it could be a part of the packages for the seed, treatment, and that can ultimately give a solution in getting the uniform crop establishment. And further research and development, because these findings are really, really interesting. This may drive further research into seed dormancy mechanisms that could lead to development of new agricultural technologies and practices that can enhance seed performance and crop productivity. As again, I want to bring it back to the Lucas presentation. It was like now possible with the tools and technologies, we can manipulate the response even at the single seed level. So having this knowledge at the whole seedling and the crop level might help us walking backwards to understand the reason for that, and tweaking at that level might help us to eliminate some of these challenges in the future. These are some of the references I used while developing this talk, and I thought it's maybe good to share with you all. If somebody want to get deep into this, you may kindly go through this presentation, these articles. And again, I would like to thank my organization, Bayer Crop Science, for allowing me to share this information, and also the International Seed Testing Association, ISTA, for giving me this opportunity. And these are the my colleagues who contributed for this work, and it's all the data what I shared today is generated in our lab here in St. Louis. With that, thank you very much, and here is my contact information. If anyone wants to have a further follow-up or understanding the science behind the seed dormancy, and we could continue to work on this area to make improvements and finally deliver a high quality products to our growers across the globe. Thank you.
Thank you as well from our side, Ganesh. That was a very nice presentation as well. In the meantime, I see that we got a number of questions, and Lucas started to answer the questions. I will probably ask them for the whole audience here. We can start with the first one. And Lucas, I would kindly ask you to rejoin here again.
Yes.
I could read your answers now, but you can also let us know what the answers are.
Yes, sure.
There was a question on how does the mutation of key transcription factors affect germination according to the germination assay shown?
Yeah, so here I just showed a brief summary of the results, but most of the mutation in terms of the germination assay really affected the germination in a way that it accelerated the germination significantly. Then regarding the phenotype of the seeds, most of them were identical. We only noticed a slight difference in color, but just to humanize, we did not measure this parameter. And then also, we also did differential genes expression on this mutant, and we saw that according to the mutant, we had some genes that were upregulated, others that were downregulated. As I mentioned in my answer, I don't have all the the numbers in my head, but I think this is in a Nature Plant paper, and in the supplementary data we have the list of all the genes that are regulated according to the mutant. So if you want to have a look and find your favorite genes, go ahead.
Yeah. Another question. Thank you for the beautiful presentation. Are you also anticipating to look at the endosperm, considering its importance in the regulation of germination slash dormancy? And if so, could you decipher using this technique uh, the communication between both tissues on a molecular level?
Yeah, that was a very nice question, and I was expecting something like this. So yeah, of course we are interested in the endosperm story, if I can say, and we are trying to focus on this. I didn't show any endosperm related data so far. We do have some. I can tell you that we've seen that the endosperm show a specific pattern in terms of transcript in our seeds that are releasing dormancy. We can see some transcript that are not here in the seeds that are still dormant. We tried to confirm some of this transcript using in situ hybridization. We saw the same pattern, it was positive. We would like to confirm this by using the spatial transcriptomic methods I presented. Then regarding the communication between both tissue on a molecular level, really in crops with, for example, in barley with a way bigger endosperm, I don't really see any problem with it. But however, in Arabidopsis, considering the layer of endosperm and its size, I'm not sure the current resolution of this technique will allow that, but everything's possible. But that's still a lot of work.
Thank you very much. I take the next one for you, and then we probably move a little bit also to Ganesh and come back to you later. Lucas, it is good to have in model plant. However, it could be different for crops. Could you explain if something is done to compare the data for Arabidopsis and field slash vegetable crops?
Yeah, that's a question we also ask ourselves quite frequently. Um, so this is really an important point. So in the lab we also work on barley, and I know some people worked on rice. So I typed this answer and I mentioned that because there is some database that are starting to flourish on the web where people are gathering the different data from the different paper, and you can import those data. And what we are doing is that, for example, we import some rice data, some barley data from other labs, from other single cell experiments. We try to find some orthologs, and we try to see if we can match and if we can find some similarity in our Arabidopsis finding. But even I do think that even in between varieties, there will be still differences also in the expression of the genes. And in the same idea, we also try to map our transcription factor to these orthologs in crops genome and examine their expression in existing crop RNA-seq data sets. There is not a lot of data available so far, and we also have to be cautious with single cell data because it's brand new technology, and now it's getting better. The paper, they show a lot of validation, but we still need to be cautious with the data to see if we can use them and if they are quite robust. So once again, still a lot of work, but the field is evolving quite fast, and I think it's quite promising and exciting.
Now, Ganesh, let us come to you with a few questions. One is, how do external stress factors, for instance, drought, temperature fluctuations, salinity, trigger secondary dormancy at the molecular level? You started to talk about that in your presentation, and I think that's a clarification which is requested
Uh,
here.
yeah, thank you for that question. Basically, some of these stressors, like abiotic stress, I would also add one more stress is cloudy days, or the weather that is um, during the late phase of seed filling. The cloudy days can also add to that. So what happens normally is, if you look at the ABA that is accumulated in the seed, The main function of ABA to avoid any kind of precautious germination on the crop. This is evolutionarily actually that trait has been integrated into the crop systems. However, the ABA level has to come down at the time of my seed moisture gets to say 10-15%. It has to come down less than 20 picomoles. That's a normal trend that we need to we see in most occasions. However, under these conditions, there is either ABA catabolism itself is reduced during the any kind of stress, or there could be a de novo synthesis of ABA because of the response to the stress. That is probably the potential reason why at the molecular level there is a difference in the hormonal regulation leading to the higher residual ABA in those seeds. At least some evidences we have is whenever there is a cloudy weather, whenever there is a high stress like situation, we have seen ABA. Besides that, there is also other hormone I mentioned, like methyl jasmonate and jasmonic acid. Those are the other factors can also contribute to this, which very little known about it because in the dormancy, you know, context.
Mhm. There were two questions on what kind of vigor test used or test is used after pre-chilling of corn. Is it cold test? If yes, what would mean additional chilling period?
Yeah, it is a cold test, cold vigor test. We use the after the chilling treatment, pre-chill treatment. So the pre-chill treatment temperatures are much lower than the the cold stress what we used in this vigor test. So the main part here is at 10C pre-chill. The main intention is to not to induce any physiological changes to the great extent, but just to trigger the system in such a way that there is a the catabolic activity of can be triggered to reduce the abscisic acid level. We could even still try at 6C. That was our probably the next approach. Team would like to see if you run at 6C pre-chill, will we get the same response or different response? So that's something in play, but definitely lower than the bigger test temperature.
Yeah. Is coming to chilling, there are two questions from the same person, I think only um, specifying one the other. What want to know pre-chilling treatment details and which kind of method used for pre-chilling, inhibitional pre-chilling or along with media pre-chilling? That is one part of the question, and I think the second half um, is no.
Yeah, I think
Actually,
yeah,
yes.
I addressed that question, I think, but yeah, it's a imbibitional pre-chilling. The seeds are planted on a in the the germination media, and then sufficient moisture is given in the media, and then the pre-chill was administered.
And before we go back to
And,
Lucas, another one.
and,
Yeah.
and just to add a point there, without without imbibition, the pre-chill effect is not seen. So that is very important. It is pre-chill is given along with the imbibition, then only the impact is seen.
And one question here, is your research based on both raw and dressed seeds?
This was study was done in the raw seed. That means that is a freshly harvested seed. If that is a question. In the finished seed, the extent is not this high. In the finished seed, because that would have lapsed, there is a time difference between the raw seed and the finished seed. So the impact is very less. So sometimes we make decision on the raw seed result. So that's where this problem is kind of encountered.
Okay, let's go back to you, Lucas. And there are a few open questions. Um, some of you answered online. Super interesting work. Thank you very much. I was wondering about your single nuclei work and the potential role of endosperm. Currently, I did not see any endosperm cells annotated in the UMAP you showed. Could you tell us something about where they went and if there is a technical reason whether you think it will be possible to extract them?
So yes, actually, there is the endosperm cells in the dataset. I just didn't display them in the presentation yet. So we find them and they are definitely there because for this experiment, I did not mention it, but because we wanted to capture the whole seeds, we optimized the protocol to work on the complete seed. We did not isolate the embryo out of the seed coat. Like this ensured we could extract all tissue and analyze the endosperm nuclei along with the embryonic tissue. So yes, the endosperm cluster are in our data set and they show distinct transcriptional profile, which is why we're particularly excited about using the spatial technique to confirm their location.
Now, a PCR question. Is it possible, Lucas, to combine single cell analysis and digital PCR for precise quantification?
Well, I'll be honest, I've spent a long time. I mean, I'm not really familiar with digital PCR precisely, and the combination of both, I haven't seen that either in commercially available technique or in publication. I think that would be maybe interesting to target low expressed genes with high precision quantification. But I also wanted to mention that this technique so far still mainly capture only the messenger RNA, and mainly they focus on short read sequencing. There is some publication, some new tools that are now doing long read sequencing. So maybe in the future, but also I would say that the technical challenge would be to integrate both techniques together. And I assume that would take some time, but everything's possible.
In addition, did you try to do single cell analysis on soybean seed, also mature seeds?
So I haven't tried single cells experiment on soybean seeds. I know there is at least 2 or 3 paper working on soybean where they use single cell technology, including one in Denmark. And one of the postdocs from this lab is now working in the same university as mine. And he's now a group leader there, Dougal Wright. So I think if you Google him, you have the answer about this work on soybean. Then regarding the mature seeds on soybean, I have no idea, but on Arabidopsis, I've never done dry seeds, if this is what you mean by mature seeds. I only focused on imbibed stratified seeds and early germination time point. Every time we change the time point, very complicated to optimize. I mean, it took some time. So dry seed, I assume, would be technically challenging for nuclei extraction because the tissue is very compact and dehydrated. And we show already, that would be interesting because we can see that there is already the epigenomic remodeling as I showed in one of my first slides. But yeah, everything would be interesting.
Okay. Let me go back to Ganesh maybe for some other questions. Need to see which you answered and which you did not. When you harvested, Ganesh, the corn seed at 40% MC and the ABA content was high, were the seeds at their physiological maturity stage?
And there's a you cut in and out. Could you repeat that quickly?
Okay, yes. When you harvested the corn seeds at 40% MC and the ABA content was high, were the seeds at their physiological maturity stage?
Yeah. Um, the question is whether that attain the physiological maturity. We see the black layer movement. So these seeds, when they hit about 35%, 40% is a upper hand, but when you hit about 30% moisture, they attain physiological maturity under most growing conditions. What happens is, because of the heterogeneity in the field, it's a large fields in a commercial production. It is very hard to precisely select a designated moisture level in the field. There could be always a range. Even we set a target of 25%, we could find some plants with 40% moisture, some with probably 25% moisture. So to account for this heterogeneity in the field, we looked at the range. However, the 40% is not a typical moisture the crop is harvested. It's around 30%. That's when we expect complete physiological maturity.
Mhm. And one very practical question to you, Ganesh. Based on the treatments you explained for breaking dormancy, I would like to ask from a practical standpoint. You,
Yeah.
you read that question before, I think. Are these treatments more feasible to be implemented at the seed company seed processing level or applied by the farmer user of the seed?
Yeah, excellent question. The pre-chill treatment, what I demonstrated, this is from the understanding purpose. This is trying to see what are all it's very effective in breaking dormancy, but practically we cannot do this. It is not possible or not feasible either at the industry level or at the growers level. The other way to do is, now we understand what are the molecular responses occurring there. We show that there is some changes in the hormonal levels, hormonal interplay. We could use those as a target molecule and bring that on to the seed as a seed treatment. Like for example, GA3 came as one of the molecule that can help eliminate this problem or mitigate this problem, that could be incorporated in the seed treatment. Okay, the next question comes, how to deliver it, what will be the cost. There's another aspect of that, but yes, and the technology will only get better as we learn more about it. So good question. Pre-chill cannot be commercially applicable because it's only from the testing perspective.
Okay. On secondary dormancy and breaking methods, what is the natural time duration to overcome dormancy in corn and any large-scale dormancy breaking methods? Now,
So
also a
we
very interesting question.
So we do at least the knowledge what we have from the some of our production location. Again, it can vary from region to region, genetics and things like that. We have seen like up to 4 to 6 week post harvesting, we will see maximum percentage of the secondary dormancy being released. If you have a slightly warmer condition during that period, it even accelerate a bit, but we store it at 10C because that's where the longevity is also important for us. So secondary dormancy is not a bad thing, but from the from the operational point of view, that is affecting business release decision. That's the reason we are paying so much attention to it. But otherwise, 4 weeks of the lag is something in circumstances which should be manageable. But in some cases, no, because in some regions where we harvest and immediately package and send it off to the growers. So in those circumstances, this is important to understand how to manage that. And breaking dormancy, we don't use any dormancy breaking treatment as such because we know by the time it goes into the field, it takes at least 3 to 4 months from the time it is harvested. So naturally, the secondary dormancy would have gone down substantially by that time
Good. We still have some time, and if you are not exhausted, I will go through a few other questions.
frame. Sure.
Uh, Now, Lucas again. I'm interested in GMO detection and distinguishing single GMO traits from stacked events. And I think that question is still going on somewhere.
Yeah, I
I
think it's the
think
follow-up
internal analysis of
to my
DNA
answer.
could solve the problem. I think they belong together.
Yeah, I think the same person answered then to my answer. I think so. It was about the digital PCR, if I
Yes.
remember.
Yeah.
So yeah, single cell analysis of DNA. could solve the problem. I'm not too sure. I'm not too sure about this, especially because I'm not really aware of a lot of single cell technique working directly with DNA, especially its long fragments, right, in comparison to the messenger RNA we have, because we're mainly working on short read sequencing. So yes, it could solve the problem, but it would be a huge amount of data. And if we're talking about DNA, we need to take into account as well the chromatin organization. So how can we take all this into account in the experiment? Which parameter to measure? So I think it's still a bit abstract for me as single cell analysis of DNA. But yeah, that would solve the problem. That would solve a part of the problem, I assume, if we're talking about the single cell and the digital PCR question here.
Yeah, another question and regarding your single nuclear work in the aspect of long storage RNA. Do you see a role for single cell genomics in seeds as well? And there's an addition to that question. Let me see. I specifically mean the protoplast method here.
The aspect of long storage RNA, do you see a role for single? Yes, I do. Yes, I do see the role here. One thing that would be interesting would be to perform the same experiment using single cell nuclei transcriptomic and single cell transcriptomic using protoplasts. And by doing the subset of genes, you could identify the stored one. However, have to keep in mind that the protoplast generation will generate a differential gene expression, so you will have to measure this differential expression also using bulk RNA sequencing on whole embryo with or without protoplast and subset that gene as well. That would be an idea. Let me see if I answered all the question here. Yeah, I do see the role for single cell genomics in seeds in stored mRNA. Yeah, I do think it's really important, and we know that at the end of seed development, we do have quite a significant amount of stored mRNA, but already pre-established the roles of the future cell types.
Mhm. Your work, Lucas, suggests that the embryonic endodermis plays a key role in the dormancy to germination transition, given that hormones like ABA, GA, and auxin are known to accumulate or be perceived in endodermal cells in roots. Did your single cell and multinomics analysis reveal evidence of hormone-related gene expression or regulatory networks specifically enriched in the endodermis during the dormancy release? I'm not sure everybody followed me during that question, but I hope you could, Lucas.
I think yeah, I think I did. I'm not sure if I will be able to answer all the questions here, but I'll do as much as I
It's
can.
not yet answered
So
this.
we did look at of marker related to hormones, including ABA, GA, and auxin. We also looked at light response marker, the one including glucogenesis, circadian clock, and also cell cycle, because we know there is no cell division. We did not find any specific enrichment in the endodermis, but I don't want to say any mistake. However, that's a huge bunch of analyses, and there's still some analysis to do in terms of enrichment and trying to associate and look at the gene ontology enrichment we can have. And we also want to do some what we call cake enrichment. So here it's not about the gene ontology, but more about the signaling pathway in which the genes we found were could be involved in. So we are looking at that. But I can answer that, but I don't think I can answer fully the question,
Okay.
unfortunately.
And another one where spatial proteomics used to establish the proteome level network.
So space. So in our work, we did not really use proteomics yet, but this is something that would be upcoming in the coming month or year to look at the proteomic level. So I am not sure if this has been done at the single cell resolution. I don't think so in seeds, or whether there is a lot of proteomics and phosphoproteomic that has been done and that identify different phosphosides that are really important in either seed dormancy, germination, and germination in seeds. I don't remember all the details, but I know I talked about it because this is in the Journal of Experimental Botany review, the expert view I presented. So I think you will find your answer in this publication.
Okay, question to Ganesh. What would be the best method of seed priming for improvement germination dry bean seed? And there in the bracket is red speckled and small white canning beans.
My expertise in that space is very limited. Fundamentally, from the from a seed physiology point of view, generating this priming approach and technique needs a good amount of insight on the how the seed was actually, what condition it has grown, what are factors that are causing for a lower performance or a lower vigor for that particular batch of seed or a type of genetics. And every genetics has I show that in the last slide, has a differential response to some of these cues. So it will be very hard to say one set of I would say recommendation for a given crop. This needs to be built and developed based on understanding the response of that particular species and the crop and the hybrid. And sometimes we may have to go tailor it to a given hybrid, because all hybrids are not equal in responding to the cue. Hope that gives some idea and insights.
Thank you for that. And I've got last question for Lucas before I answer the really last question. And I kept that because I think it's a very good question. Do we still believe that endosperm is a dead tissue?
So I do not have the answer I typed, but I know what I typed. Um, and I think I said something like, I've seen this fight with my eyes. I've heard people saying that uh, spatial transcriptomic is not robust, and the signal we detect in barley endosperm is not a true signal of gene expression. And I've seen people convinced that it is. And I know people were convinced already, and I think now indeed we have more and more proof. So my belief, I would say, is yes, I do believe that the endosperm is not a dead tissue, and it plays an important role. And if you want to look at a nice example of spatial transcriptomic on barley, there is a publication from 2023 from Latrobe that shows there is a lot of movement, aquaporine, and so on, that are genes related to aquaporine that are expressed in the endosperm. So it's spatial transcriptomic. It's a PNAS paper from Peyrat Lobe 2023. If you want to have a look, it's pretty good, and you'll have your proof for yourself. You'll be convinced.
Yeah, thank you for that. I kept the last question for me, not because it's a technical question. It's more a question on administration. The question was, can we have recording of this session? And yes, there is a recording of this session available shortly on the ISTA YouTube channel. At least you can follow that up there and subscribe also to the ISTA YouTube channel. Then you get all the information when they are available. We are very good in the timeline. I have no other questions. I've got a lot of other remarks in the question and answer area, which are thank yous. And thank you very much for the brilliant talks and your answers to them. Uh, I can only say the same. Thank you very much for both of you. It was very interesting, and the reaction of the audience, where we had more than 250 people and the maximum time online, that was really great. Thank you, and have a good day. And thank you to everywhere where you are in the world and listening. Don't forget, you can get uh, go to the ISTA YouTube channel in a few days and find uh, the presentation there. Thanks a lot and have a good day. Thank you very much.
Thank you, Andreas.
Thank you.
Nice meeting
Bye.
you.